not entirely human. In fact, 8% of your DNA originally came from viruses. Millions of years ago, ancient viruses infected our mammal ancestors, leaving molecular fossils buried deep inside our genetic code. You are a walking graveyard of prehistoric plagues. But today, we aren't just worried about the viruses of the past. We are terrified of the viruses of the future. Look at the news. People are asking the same chilling questions. What if a virus escapes from a high-security lab? What if they are genetically modified and used as biological weapons? What happens when the next big one gets loose? These are not just movie plots. Viruses have escaped from research labs in the past. And today, scientists possess the actual technology to genetically modify viruses. Researchers have already used reverse genetics to literally resurrect the deadly 1918 pandemic flu virus using DNA pulled from victims buried in frozen permafrost. The threat is real. But to understand what viruses could do to us tomorrow, you have to understand how they actually work, how they evolve at terrifying speeds, and the brutal microscopic war raging inside your body right now. Let's start with the basics. What actually is a virus? By most biological standards, viruses aren't even alive. They're the ultimate biological gray area. They have no metabolism. They cannot respond to stimuli. They have no machinery to generate their own energy. A virus takes simplicity to absolute extremes. At its core, a virus is usually nothing more than a tiny string of genetic material, DNA or RNA, wrapped inside a protective protein shell. Sometimes that shell is covered in a lipid membrane. That's it. Because viruses lack their own machinery, they are completely helpless on their own. They cannot reproduce. They are entirely parasitic. They must find a host. When a virus comes into contact with a host cell, it injects its genetic code inside. The host cell is tricked. It unknowingly reads this foreign code as if it were its own. Instantly, the cell stops its normal functions. It is hijacked. The cell transforms into a virus-making factory. It cranks out hundreds, thousands, sometimes tens of thousands of new viruses until it is completely destroyed. When the cell bursts, all those new viruses spill out to hunt down more cells. Scientists still debate where viruses came from. Some think they're ancient relics from an RNA world, predating life itself. Others think they escaped from our own cells, millions of years ago. A third theory suggests viruses were once complex cells that devolved into parasites. But one thing is certain. Viruses have been shaping life on Earth since the very beginning, and they are evolving faster than we are. Viruses are the fastest evolving entities in biology. Here's why. Every time a virus hijacks a cell and copies its genetic code, mistakes happen. Think of it like a typo in a massive manuscript. Most of these typos are fatal for the virus. They break something essential. But every once in a while, a random mutation changes a viral protein in a way that gives the virus an advantage. Maybe it helps the virus replicate faster. Maybe it helps it hide from the immune system. The speed of this evolution depends heavily on the virus's design. DNA viruses, like herpes or pox viruses, copy themselves using host enzymes that literally proofread the code. These enzymes catch mistakes and fix them before they matter. Because of this, DNA viruses mutate slowly. They're relatively stable. But RNA viruses like HIV, Ebola, influenza, and coronaviruses are different. They use enzymes called RNA polymerases. And here's the crucial part. These enzymes do not proofread. They are sloppy. They make millions of mistakes. Every single time a virus replicates. Because of this, RNA viruses mutate at a terrifyingly high rate. This is what makes HIV so incredibly difficult to cure. Let me show you exactly how this works. When a patient takes a single antiviral drug, the drug wipes out most of the HIV virus. Most, but not all. See, because HIV mutates so fast, the virus population inside a single patient has massive genetic variation. You might have billions of copies of HIV, and each one is slightly different from the others due to random mutations. So when the drug attacks, it kills most of them perfectly. But by pure random chance, a tiny fraction of the viruses will possess a mutation that makes them completely immune to the drug. These survivors are now safe from the medication. They replicate, they bounce back, and suddenly, the entire viral population inside the patient is drug-resistant. This is why doctors cannot use a single drug to treat HIV. The virus will always find a way around it. Instead, doctors use something called HAART, a highly active antiretroviral therapy. It's a cocktail of 3 or more different drugs, taken at the exact same time. Why does this work? Because it's statistically nearly impossible for a single virus to randomly develop resistance to 3 completely different drugs at the same time. It would need 3 separate beneficial mutations to occur in the same virus. The odds are astronomically low. This is evolution in real time. Humans fighting back against a virus that evolves faster than we can possibly imagine. But mutation isn't the only trick viruses have up their sleeve. They have something even faster. When 2 different viruses infect the exact same cell at the exact same time, things get chaotic. Inside the host cell, the genetic material of both viruses is floating around. Sometimes, frequently actually, the viruses swap whole chunks of their genome to create a completely new hybrid virus. They're sharing genetic code like 2 programs being merged into one. This is called recombination, and it's how some of the most devastating viruses in history were born. Take SARS-CoV-2, the virus that caused the COVID-19 pandemic. SARS-CoV-2 shares heavy similarities with a bat coronavirus. But here's the strange part. Its critical receptor-binding proteins, the part that lets the virus stick to human cells, look almost exactly like a virus found in pangolins. 2 ancient viruses likely met inside a single animal. They infected the same cell at the same time. They swapped genetic code. And what emerged was a hybrid capable of infecting humans. Think of it like nature's way of genetic engineering. Random mutation is like rolling the dice once. But recombination is like shuffling an entire deck of cards and dealing a completely new hand. It's how viruses pull off the seemingly impossible. Jumping from animals to humans and spreading across the globe in days instead of years. One animal, one cell, one moment of genetic mixing. And suddenly, we have a pandemic. But humans aren't defenseless. Inside you right now is the second most complex biological system known to science. Your immune system. To understand the scale of the war happening inside you, you have to realize something fundamental. You are not a single person. You are a massive, walking planet made of roughly 40 trillion individual cells. For a virus, your body is a warm, resource-rich ecosystem. It's an all-you-can-eat buffet. The problem is speed. A bacterium can duplicate itself in half an hour. A virus can multiply into the billions within days. Meanwhile, humans take years to reproduce and mature biologically. We are vastly too slow to keep up with their evolution. So how are we not already dead? The answer is brilliant. You actually have 2 immune systems. The first is your innate immune system. It uses general-purpose soldiers to fight common threats. It's your first line of defense. But the second is your true superweapon, the adaptive immune system. And it uses a cheat code so elegant it sounds like science fiction. Your adaptive immune system has the largest reference library in the universe. It literally has an answer to everything. Right now, floating inside your blood, you have a specific cell designed to fight the Black Death. You have a cell designed to fight coronavirus. You even have a specific cell perfectly designed to fight a deadly virus that hasn't even evolved yet. A virus that won't appear until a city is built on Mars 100 years from now. How is it physically possible to have a defense for threats that don't exist yet? It comes down to protein puzzle pieces. Every organism and every virus is made of proteins, folded into billions of unique 3D puzzle shapes. To fight a virus, your immune cells need to bump into it. touch its protein puzzle piece, and recognize it as an enemy. To be prepared for billions of possible mutations, your adaptive immune cells are given official biological permission to deliberately mutate their own genetic code. They take tiny fragments of their own DNA and mix them together completely at random to create unique receptors. They're generating diversity intentionally. By randomly mixing just a few pieces, you can create billions of unique recipes. Your cells do this with their genes, generating up to 10 billion unique immune cells, each holding a completely unique receptor. You literally generate a lock for every possible viral key in the universe. But this creates a terrifying problem. If your body is randomly generating weapons designed to attack every possible protein shape in the universe, it will inevitably generate weapons that perfectly match your own organs. Your immune system will accidentally generate assassins designed to murder your heart, your lungs, your brain. To prevent this catastrophe, your body funnels every single one of these newly forged superweapons into a specialized organ sitting right above your heart, the thymus. The thymus is essentially a microscopic murder university. Inside the thymus, your body forces these young immune cells through an intense, deadly curriculum. Teachers present protein puzzle pieces taken directly from your own body. If a young immune cell attacks one of your body's own puzzle pieces, the teacher orders the cell to immediately commit suicide. It is killed, eaten, recycled. The immune system is so brutally strict about this that 98% of the cells that enter Murder University are executed. Only 2%, the cells that prove they will not attack you, are allowed to graduate and enter your bloodstream to hunt viruses. When this process fails, when those assassins escape, you get devastating autoimmune diseases like lupus or rheumatoid arthritis. This brutality is why you don't have autoimmune disease. This is also the frontier of the invisible war. The invisible war never stops. It's an arms race, and the viruses fight back viciously. Take HIV. Your cells evolved a brilliantly aggressive defense protein called Tetherin. When an infected cell tries to release new viruses into the bloodstream, Tetherin acts like glue. It physically locks the newly born viruses to the surface of the cell so they can't escape and spread. It's a cage, a prison. The virus is born, but it's immediately captured. For millions of years, this worked. In monkeys, the simian immunodeficiency virus evolved a protein called NEF to break this Tetherin lock. NEF could grip Tetherin and destroy it, allowing viruses to escape. But when SIV spilled over into humans, it hit a wall. Human Tetherin has a slightly different shape than monkey Tetherin. The monkey virus's NEF protein couldn't grip it. It was useless against us. The human immune system was winning, but the virus didn't give up. So HIV did something remarkable. It evolved a completely new weapon. HIV-1 developed a brand new protein called VPU. VPU ignores the old lock entirely. It doesn't try to use the same strategy. Instead, it attacks human Tetherin at a completely different weak point, from a completely different angle, using a completely different mechanism. And suddenly, the virus escapes. It spreads. It takes over again. This is the invisible war. Every mutation the virus makes is matched by an adaptation from our cells. Every adaptation our cells make is countered by a new mutation from the virus. Neither side ever wins. Both sides just keep evolving. But we are finally learning how to break this cycle. So why does all of this matter to you today? Because we are no longer passive observers in this evolutionary war. We're stepping onto the battlefield. And that comes with incredible risks. The technology to genetically modify viruses absolutely exists right now. Virologists use a technique called reverse genetics. By cloning viral genomes into plasmids, scientists can deliberately design specific mutations, delete genes, or add completely new traits. Then, they insert that modified code back into cells to grow live, customized viruses. This technology is incredibly powerful. As I mentioned earlier, scientists used reverse genetics to read the DNA of the 1918 Spanish flu, a virus that killed 50 million people, and physically brought it back from the dead. But what about COVID-19? Was it an engineered weapon? While extraordinary claims require extraordinary evidence, the scientific consensus strongly points away from a bioweapon. The genetic fingerprints of SARS-CoV-2 show the messy, chaotic hallmarks of natural evolution. It looks exactly like a virus that naturally recombined between a bat and a pangolin. The evidence is overwhelming. However, the question of a lab escape is entirely different. It is an absolute historical fact that dangerous viruses have accidentally escaped from research facilities before. While the exact origin of COVID-19 remains heavily debated, the reality is this. The more we study viruses in high containment labs, the higher the mathematical risk of an accidental release becomes, and the threat of natural spillover is greater than ever. There are over 7.5 billion people on Earth. We constantly interact with animals, keeping them as pets, farming them, destroying their habitats. Every single interaction is a roll of the genetic dice. Add in the reality of global international flights, and a virus that spills over in a remote village can be in the center of New York City in under 24 hours. The conditions for explosive viral evolution have never been better. The threat is real, but so is our response. So, is humanity doomed? Are we just waiting for the next microscopic apex predator to wipe us out? Not exactly, because we are starting to learn how to manipulate the rules of viral evolution to our advantage. The most powerful thing we've discovered is this: we can actually force viruses to evolve to be less deadly. Viral evolution is a balancing act. When a virus is in a crowded environment where it can easily jump from person to person, natural selection favors the fastest, most aggressive viruses. They replicate wildly, often overwhelming and killing the host. But it doesn't matter, because the virus has already jumped to the next victim. But if we use physical barriers like masks, hand washing, and social distancing, we change the environment entirely. Suddenly, the virus is trapped. If an aggressive virus kills its host too fast now, it hits a dead end. It goes extinct. It dies without spreading. Under these conditions, natural selection forces the virus to mutate into a slower, milder, less deadly version. By simply slowing the spread, we literally drive the evolution of the virus in our favor. We are turning their own weapon, evolution, against them. We're also learning how to weaponize viruses for our own survival. When viruses are grown in laboratories in petri dishes, they often undergo something called gene reduction. In the wild, viruses carry complex genes designed to fight off human immune systems. But in a cushy petri dish with no immune system to fight, viruses simply mutate and delete those genes. They don't need them anymore. We use these defanged, mutated viruses as live, attenuated vaccines. They teach your immune system to recognize the virus without being able to cause serious disease. And it gets better. We are now hijacking viruses to cure human diseases. Using reverse genetics, scientists are taking massive DNA viruses, gutting their harmful genes, and packing them full of healthy, normal human DNA. These harmless recombinant viruses are then injected into patients suffering from genetic disorders. The virus does what it does best. It breaks into the patient's cells. But instead of delivering a payload of death, it delivers the cure. This exact method is already being used in gene therapies for diseases like cystic fibrosis and severe combined immunodeficiency, SCID. Patients who would have died are now alive. And it's all because we learned to reprogram the enemy's weapon. We're even using recombinant vaccinia viruses, the same viral family that eradicated smallpox, as Trojan horses. Scientists insert the DNA of cholera or rabies into the harmless vaccinia virus. When injected into your arm, the virus trains your adaptive immune system to fight off diseases you've never even been exposed to. It's like sending a messenger into enemy territory carrying a detailed blueprint of the enemy's face. Your immune system sees it, learns it, and prepares to fight. Viruses are the ultimate shapeshifters. They are ancient, invisible, and utterly relentless. They will continue to mutate. They will continue to shuffle their genes. They will continue to spill over from the animal kingdom into us. The threat of a new pandemic, whether from nature, a lab accident, or a rogue actor, will never fully disappear. But for the first time in the billions of years of life on Earth, one species is finally fighting back. We are reading their code. We are tracking their mutations in real time. We are turning the greatest parasites in biological history into our greatest medical tools. The invisible war is far from over, but the tide is starting to turn. If this video blew your mind, hit that like button, subscribe to Evolution of Anything, and turn on notifications so you never miss what we uncover next.